Welcome to part 5 of the nervous system. Today we will explore the interactions with the muscular system. Our enduring understanding is 4A, 4B, interactions among systems. We'll see interactions and coordination between systems provide essential biological activities. The illustrative example we're going to use is the nervous and muscular system interactions. So when we look at a simple contraction of a muscle, this typical response generated by the nervous system, it demonstrates or illustrates for us the interdependence of the nervous system and the muscle systems, how the two work together to coordinate movement. As we consider the interaction of the muscular and nervous system, let's look at how a muscle fits into the hierarchy. When we begin to think about how a contraction occurs, we need to know all of the parts that are involved in that process. So we have the organ, the muscle, which is made of bundles of fibers, in this case muscle fibers. And the muscle fiber bundles are actually bundles of individual cells, long, thin, multinucleated individual muscle cells. And so the organ is the muscle, and then the tissue in this case are the muscle fibers, and then each single muscle fiber is an individual cell. Now within that cell, we're going to see these repeating bands of sarcomeres. So here's one, then you can see another repeating little segment. It makes the muscle appear segmented or striated as you look at it. So there are areas where there's light and then it gets a little dark and then light again. So from this band to here, from Z line to Z line, this part inside a muscle cell is called a sarcomere. And a sarcomere is the basic contraction unit when we are talking about how a muscle actually works. So here's an electron micrograph that shows you a sarcomere. So here's one Z line, here's another Z line, and then there's an area that's got a lot of the myosin protein, and then the band called the I band has much more of the thin filaments. So this is a diagram of the same thing. You can see it a little bit better. So here's where we'd have one Z line, here's the other Z line. And notice that the thin filaments are what actually form the Z line. And what happens in a muscle contraction is that this piece of the muscle cell is going to slide across the thicker piece inside here. So the thin filament is going to be contracted this way. Over on this side, this thin filament is moving this way. So we're going to move the two sides closer together and fill in this M line that's in between. So the thin filament will be pulled toward the M line or the midpoint of that sarcomere as a contraction occurs. So those are a lot of words. Let's take just a minute to organize this in your brain. The parts that we've just mentioned. You should be able to arrange these components into a structural hierarchy from large to small. Think about the words that are listed there on the left and put them in order from large structure, what's the largest in this list, and then arrange them down to what's the smallest. You'll probably want to stop the screencast for a moment to do that. Hopefully you have arranged them in this order, with the muscle being the organ. It sits at the top of our list because we're down inside the muscle system, so we would look at the muscle organ. Then there is the tissue level when we are talking about muscle fibers. That's the tissue level. And then a muscle cell, of course, is at the cell level in the hierarchy. The myofibril are sets of repeating sarcomeres, and sarcomeres are structures inside the individual cell, and then they are composed of a thick protein called myosin, and then a fairly thin protein that is called actin. And so that's the structural hierarchy of a typical muscle contraction unit. So when we look at a muscle contraction, do you notice the difference in the distance between the Z-lines when a muscle is relaxed, as opposed to the distance between the Z-lines when the muscle is contracted. The actual muscle doesn't stretch or expand. It actually just shifts position where the thin filaments move from being far away from the thick filaments to moving closer, so that this gap is closed. So this light area is gone in a contracted muscle because the thin filaments have moved across the thick filaments. And that's what makes muscle contraction occur. When this thin filament, that's right here in yellow, moves closer to the inside or closer to the M band, that's right in the center, then we would say that that muscle is contracting. Here, this muscle's sarcomere is completely expanded or extended, and this muscle would be in a relaxed state. Notice there's this shortening of the distance between the thick filament and the Z band as that muscle contracts. So kind of like stacking cards on top of each other. The thin filament slides across the thick filament during a typical muscle contraction. So now we're going to look at muscle contraction on a molecular level. I'm going to show you a video clip that describes the process. You may want to repeat the clip several times in order to get the main idea.
Skeletal muscle is composed of bundles of elongated muscle cells called fibers. Each muscle fiber or cell contains thread-like structures called myofibrils. A myofibril is made of units called sarcomeres, joined end-to-end by Z-lines. Each sarcomere contains both thin and thick strands of proteins known as myofilaments. The thin myofilaments are attached to the Z-lines at both ends. Thick filaments are grouped in the sarcomere's center. When thin and thick filaments interact, they slide past one another. This sliding action pulls the Z-lines toward the center, and the sarcomere shortens. As the sarcomeres shorten, the entire muscle shortens, or contracts. Thick myofilaments are made of the protein myosin. Individual myosin molecules have a two-part head and a long tail. A myofilament may contain 200 or more myosin molecules bundled together. The tails form the center of the bundle. The globular heads project outward and in opposite directions at each end. These projections, called cross bridges, have hinges that allow them to bend during muscle contraction. Thin myofilaments contain 3 different proteins. Actin, the primary protein component, has 2 rows of molecules coiled into a helix. The second protein is tropomyosin. These long, narrow molecules wind along the grooves between the actin. The third protein, troponin, is a complex of 3 subunits. Troponin is bound to tropomyosin at regular intervals along the groove. Together, troponin and tropomyosin control actin's ability to interact with the thick myosin filaments. Contraction begins when nerve impulses arrive at the muscle cell. This stimulates a membranous network of channels called sarcoplasmic reticulum to release calcium into the myofibrils. Calcium binds to troponin, which causes tropomyosin to move, exposing the active sites on the actin molecules. The myosin cross bridges attach to the active sites. The binding of the myosin cross bridge to actin triggers the release of stored energy. This produces a force that causes the cross bridges to bend. As they bend, the cross bridges pull the actin molecules toward the center of the sarcomere. This pulling action is called a power stroke. For contraction to continue, ATP must bind to the myosin cross bridges so they can detach from actin. The splitting of ATP energizes the myosin cross bridge, thus preparing it for another power stroke. Each power stroke pulls the actin closer and closer to the center of the sarcomere. As the myofilaments slide, they continue shortening the sarcomere, and the entire muscle contracts. Contraction ends when nerve impulses cease. Calcium is actively transported back into the sarcoplasmic reticulum away from the myofibrils. In the myofilaments, the troponin tropomyosin complexes cover the active sites. Although the myosin cross bridges are energized, they cannot attach to actin. In the absence of calcium, the cross bridge cycle ceases and the muscle relaxes.
So that's a lot of information to take in at once. So let's break it down into steps. We can put this action into 5 steps, and we'll look at them one at a time. In the first step, let me just remind you that there are thin and thick filaments present in a particular sarcomere. So there are thin filaments, or the actin filaments, and then there are the thick filaments that have the myosin heads on them. And so the thin filament and thick filament are going to slide past one another, and as they do, the muscle will shorten or contract. It's interesting to note that in order for the myosin head to let go, we have to spend ATP. So ATP is involved in each muscle contraction that happens. So when the myosin head is in this configuration as shown here, this is the high energy configuration, and the myosin head is able to connect to the actin filament and form that cross bridge. Once the cross bridge is formed, then we have the power stroke is going to take place as ATP is consumed, and that thin filament will move toward the center of the sarcomere. The myosin head is now in its low energy configuration, and more ATP will have to be spent in order to let this myosin head let go of the thin filament. And so it requires the input of ATP in order for that cycle to continue. So each time a muscle contracts, we're going to have the need for using energy or ATP. So our muscles tend to be energy gluttons and take in a lot of energy. But remember, in the video it indicated that this whole process is started when a stimulus is received from the nervous system. The next animation just runs up here in the top corner. You can see it. And notice here is the myosin complex. You can see that it's sort of pulling and continuing to work as ATP attaches and triggers that power stroke to let go, and then the thin filament can be pulled or slide across more directly toward the thick filament. And notice that there is calcium as well as magnesium and phosphorus involved just in the simple act of contracting a muscle. So there's a lot of synchronization here. We see this process is highly regulated. The amount of calcium that comes in actually controls this process, and then also the removal of that calcium in the end when it's actively transported back out and the stimulus stops. So here you see the tropomyosin, and along the tropomyosin occasionally you'll see the calcium binding sites, so that when the nervous system stimulates the release of calcium, calcium will come in and bind with at the calcium binding sites. And when that occurs, you'll notice that the myosin binding site here, these little dark areas on the actin, when they are exposed, then that myosin head can attach. And so in that way, calcium is impacting or regulating the ability of this muscle to contract. So as the neuron comes toward, or as the impulse comes down the neuron toward the muscle, then this interaction or the interdependency between the nervous system and the muscular system is exemplified as this sarcoplasmic reticulum stimulates the release of calcium. And then when the calcium is present, then that is going to cause those sites on the actin to be exposed, because the calcium binds with the troponin and exposes those binding sites so that the contraction can occur. Now, we don't have all of your muscles contracting at one time. Not even inside a single muscle will all of your myofibrils contract at once. In fact, if you'll notice here, this nerve that's indicated by the blue coloration touches some of the muscle fibers, while the green touches different ones. And so this motor unit involves the muscle cell 1, 3, and 4, while the nerve that's feeding the motor unit number 1 here, colored in blue, involves the muscle cell number 2 and muscle cell number 5. So if only motor unit number 1 is working, I'll get a less contraction than if both 1 and 2 were working. So in that way I can have a graded contraction or a shorter contraction or longer contraction based on the amount of motor neurons that are involved in the contraction effort. So that concludes our look at how muscle contraction works on a molecular level. It is highly integrated with the nervous system, and I think you'll agree that that's a fairly detailed process, so it may warrant you repeating parts of this video cast to make sure that you understand it completely. And then the next time we will look at sensory reception, specifically at the eyes and the ears.